Hi! Sébastien here! Today I will show you how to visualize a sample from the Human Microbiome Project using Ray Cloud Browser. Just to put you in context, what is this microbiome project? This project is an effort to analyze the bacteria that are on our body and inside our body. This is hard to do because a lot of these bacteria are hard to grow in a laboratory. This process of analyzing these bacteria is done by sequencing their DNA. Sequencing their DNA is performed using what we call DNA sequencers. Here is a description of the sample we're going to visualize. The sample is SRS01134. This sample was generated using DNA from human stools. This DNA is presumably mostly from bacterial source, and we have a bunch of files on a computer containing the sequences. How large is this data set? It's about 13 gigabytes and it's compressed. I will show you how to visualize this data using Ray Cloud Browser. We have to select the sample. After that, we select a section. There's just one here. And finally, I select a place where we want to start, and we simply go there. The blue moving squares located in the top of the display tells us that it is loading. Now that it is loaded, I will explain what we see. The circle with a letter inside, A, T, C, or G, represent DNA sequences found in the sample, and the links between them are genetic overlaps of all but one letter. These letters are called nucleotides. The colored paths we see were computed by Ray, which is an assembly software for genomic analysis. Hovering over a circle shows us its true sequence in the bottom left corner. Also, the life bar on top of each circle represents its abundancy in the sample. What can I do with that viewer? I can explore huge DNA graphs using a web browser. That's cool, is it not? For example, I can select a part of this graph to inspect its DNA sequence. I can also zoom in and out and move around using the controls. What else can I do? For me, the main entertaining feature is to press the play button and let the thing transport me. That's nice, but where is the science? With this tool, I can inspect the structural relationships between DNA sequences found in a given sample. For example, here we can see one repeated region unwind into numerous strings that use it. Let's resume our journey in this part of the graph. We have now reached a dead end in the graph. A dead end means that at this location, there is no more sequencing data available. Pressing the graphs button shows the sequencing depth at each position in the path. With this, we can see the drop at the end. A dead end can also mean that the assembly software went the wrong way. I showed the end of a DNA path in this graph with Ray Cloud Browser. Most of the paths are easy to find in the graph because a sizable part of this graph is just long linear stretches of DNA. Let me show you an example. As you can see, this is easy to go from here to there because there is just one way to do it. This completes what I wanted to share with you today. Thank you for watching and happy visualization of your DNA data.